So this lesson is on plant tissues. Okay, so in order to understand plant tissues, we need to first understand what is a tissue and what is an organ.
So a tissue is a group of similar cells that carry out the same function. Okay, so it's basically all the same cells that are doing exactly the same job. Okay, an organ is a group of different tissues that work together to carry out the same function. So, for example, a leaf, okay, there are lots of different tissues, lots of different types of cells that all work together to do certain jobs. So we have a look at the structure of a leaf now. Okay, I'm just going to put these in. So, starting at the top, we've got the upper epidermis, okay, with the waxy cuticle. This acts much like the skin in humans and animals. Okay, the job is to provide a physical barrier that prevents physical damage and things like viruses and bacteria getting into the leaf.
It also reduces the rate of water loss.
Okay, next layer down is the palisade mesophyll. This is where photosynthesis mainly happens, okay? These cells are tall and thin and are packed with chloroplasts so that they can maximise the amount of photosynthesis that can be done in the leaf.
They are also packed closer together and they are towards the top of the leaf.
The next layer down here, the spongy mesophyll, okay, this is where gas exchange happens, okay?
The cells here are packed loosely for efficient gas exchange.
The gas will come in and out of the stomata, okay, we'll get to that in a minute, and this is where the gas exchange happens. The lower epidermis acts exactly the same as the upper epidermis. It has a waxy cuticle as well. The only difference is you'll find stoma, which is one, or stomata, which is more than one, dotted throughout the underside of the leaf, okay? The stomata have got guard cells next to them either side, okay? We're going to look at them in a bit more detail in a minute, but through this hole, that's where the gas exchange takes place.
Then you've got the xylem and the phloem, which together are known as the vascular bundle. Okay, these act much like veins and arteries in animals. Okay, the xylem carries water and minerals around the plant, and the phloem carries the sugars and everything else.
So, have a closer look at guard cells and stomatas, okay? So this is what a stoma will look like when it's open, okay? The guard cells are sausage-shaped cells that flank either side of the stoma. Okay, when the plant wants to take in more gases for gas exchange, they will open up like this, okay? When the plant doesn't want to lose water and doesn't want to exchange gases, okay, they'll close up like this, meaning that much less water will be able to escape.
Okay, so this is a microscope image, a light microscope image of stomata. Okay, just to recap, okay, the main role of the stomata and the guard cells is to control gas exchange and water loss. Okay, the stomata are pores on the waxy cuticle of the leaves. Okay, you mainly find them on the underside, but you do find them on the top as well, but in much smaller numbers. Okay, the guard cells surrounding each stoma help to regulate the transpiration by opening and closing the stomata. Okay, light is the main trigger for the opening or closing of the stomata. And they do this, okay, when there's light, the guard cells absorb water by osmosis. That means they become turgid and the stomata opened, okay? So they go more banana shaped, okay? And it opens up the guard cells, opens up the stomata, sorry. When it's dark, the guard cells lose water through osmosis and they become more straight, okay, which closes up the stomata.
A way that we can see the stoma, the stomata, and count the stomata, okay, is we can paint the underside of a leaf with clear nail varnish, then use a piece of sellotape to peel it off, okay, so you'll end up with something that looks like this. These are the stomata. Okay, then what you want to do is stick the tape onto a slide and then view under the microscope. You could just view the leaf directly under the microscope, okay? There's two ways of doing this. What we do then is count the number of stomata. You then put this into a table. Okay, you repeat it however many times more that you'd want to. Okay, the more times the better, in different locations, listing the number of stomata that are counted. So we've got some typical sort of exam questions here. So we've got your information that you get here. Okay, plants lose water through the stomata in the leaves. The epidermis can be peeled from a leaf. The stomata can be seen using the light microscope. The table below shows the data a student collected from 5 areas on one leaf. Okay, so this student has repeated it 5 times, okay, then worked out the mean, the average. Okay, this is on the top, the upper surface, and this is on the lower surface. Okay, as we know, there's much more stomatal openings on the lower surface. So describe how the student might have collected the data. What is the medium number of stomata on the upper surface of the leaf? Calculate the value of x in the table. Follow the rounding pattern for the other results in the table. The image shows part of the surface of a leaf. Calculate the number of stomata per millimetre squared of this leaf surface. Okay. So to describe how the student might have collected the data, you can do it in one of two ways. Okay, you could either peel the epidermis from the leaf, okay, and put that directly, place the epidermis on a slide, count the stomata in one area, and then repeat, obviously 4 more times in this case. Or we could do the way that I mentioned a few minutes ago. You could use the clear nail varnish with the sellotape and then put the sellotape onto this slide, okay, and then do the same way, count the stomata, repeat it multiple times. To calculate the mean, okay, the mean is the average, okay, so you'd add all these numbers together and divide it by 5, you'd add all these numbers together and divide it by 5. So what is the median number of stomata on the upper surface of the leaf? So we're looking at this one here. Okay, you put them all in a line like this, so you go 0, 1, 1, 3, 5, and then the median is the middle number, so the answer would be 1 in this case. To calculate the value of x, okay, that's the mean, I've just told you, that you add these numbers together and then divide them by 5, which would equal 41.2. You round it down to 41, so you'd have 41 in this area. The image shows part of the surface of a leaf. Calculate the number of stomata per millimetre squared of this leaf surface. Okay, so first of all we need to convert it to millimetre squared. Okay, so we times 0.1 times 0.1 gives us 0.1 millimetre squared. Next we want 1 whole millimetre squared rather than breaking it down. The way we convert this number into 1 millimetre squared, okay, is we times it by 100. So if we times this number by 100, it equals 1. So because we've timed it by 100, we need to times the amount of stomata in that area by 100. Because there's 2 stomata here, we need to times 2 by 100, which gives us the answer 200.